Good afternoon, everyone, and welcome to the Infectious Disease Epidemiology Seminar Series brought to you by the Center for Communicable Disease Dynamics and the Harvard T.H. Chan School of Public Health. I'm Bradford, and I will be your moderator for this session, and I am excited and honored to welcome Professor Andrew Reed as our speaker today, who is at the Center for Infectious Disease Dynamics at Penn State. So first, let me introduce Andrew. Andrew Reed is an evolutionary microbiologist. Originally from New Zealand, he obtained a Doctor of Philosophy in Evolutionary Biology at the University of Oxford. He has held various fellowships at Oxford and then at the University of Edinburgh, Scotland, where he became Chair of Natural History. He's authored more than 250 peer-reviewed publications, 30 book chapters, and 4 edited volumes, and has been elected to fellowships from the Royal Society of Edinburgh, the Institute of Advanced Studies in Berlin, the American Association for the Advancement of Science, the American Academy of Microbiology, the Royal Society, and the American Academy of Arts and Sciences. In 2007, he moved to the Center of Infectious Disease Dynamics at Pennsylvania State University, where he is the Evan Pugh University Professor in Biology and Entomology, the Eberly Professor in Biotechnology, and Director of Penn State's Huck Institutes of the Life Sciences. His talk today is entitled Pathogen Evolution in a Vaccinated World. Thank you very much for joining us, and please take it away, Andrew.
Okay, thanks very much, Bradford. I really appreciate the opportunity to talk here. How's that looking? Okay, thanks very much for the invitation to speak here today. What I want to do is to talk about 3 separate areas we've been working on over the last 10, 12 years, all of which I think have some implications for the future, how we might think about the future of the SARS-CoV-2 ongoing evolution. But obviously, we've got to be careful about how we extrapolate. So I'm going to pitch these 3 stories in the direction towards COVID, and then towards the end, just wrap up and say what I think that might mean, what they might mean. And the first of these stories I want to talk about is myxomatosis in rabbits. And I absolutely realize that there's no vaccination involved, at least not in the work we're doing, but it is associated with changes in host resistance. So I do want to just make that point from the outset. So myxomatosis or myxomavirus spillover is a classic, perhaps the best studied before COVID, spillover event in virology. Those of you who don't know, the Australians imported in their wisdom rabbits into Australia towards the end of the 1800s to establish a fur trade that succeeded beyond their wildest beliefs. And the rabbits did what rabbits do, and they took over and turned vast swaths of Australia into desert, and it became extremely unfarmable. So the Australians through the 20s and 30s and 40s discussed a lot of different biocontrol methods, and in the end, myxomavirus was chosen and released into Australia around the Christmas of 1950. And that virus then spread like crazy over the whole of the southeastern part of Australia and killed tens of millions of rabbits, getting up to perhaps 100 million rabbits in that first summer. That is the single biggest vertebrate disease catastrophe or success, depending on your viewpoint, in that's known in anywhere. Now, to give you a sense of what happened was that had catastrophic effects on the population density of the rabbits. And since then, the rabbit population has stayed a lot lower than it was. Over the last second half of the 20th century, it increased, and the Australians released a different virus, rabbit hemorrhagic disease virus, which also had very significant impacts on the rabbit population, and that rabbit population is rebounding slowly from that. The myxomavirus release saved, is estimated, over $1 billion for Australian agriculture, so one of the most cost-effective biocontrol measures ever. When it happened, Frank Fenner, who was at that point one of Australia's leading microbiologists, pivoted his entire career to studying, I shouldn't say the entire career, but he did very significant work on smallpox later. But at this point, he pivoted his career to following what was going to happen, because if you release a virus that is that virulent, he reasoned, something ought to change. And it was killing, the initial virus was killing in the laboratory 99.9% of rabbits. And so he reasoned that it just simply had to be be such that it would evolve towards reduced virulence. And so he started taking viruses from the field and putting them into laboratory rabbits to watch the change in the death rates and the disease syndrome through time. And he was really established a fundamental research program doing this. He and his colleagues worked through the 1950s on this, monitoring the changes in virulence in the standard lab rabbit. His graduate students and colleagues took over in the 1970s. And then, as I'm about to show you, we took over in the 1990s and in the 21st century. What he found right off from the get-go was that there was a very rapid change in the virulence, initially quite slight, but over the next few years, very rapid changes, very significant changes. And the virus became less virulent. Although remember, we're talking a case fatality rate of 99.9%. So in a sense, it could only go down. And in a real sense, it's got a long way to go down before it becomes as into the case fatality rates that are more typical of human diseases. And the data that he gathered and other colleagues on why the virulence went down is typified by this figure. So in COVID speak, these are three separate strains or variants, the original variant, a less virulent variant, and a benign variant in those three colors there. And you can see that the original one kills all the rabbits and it kills them within 10 days. And so the transmission to mosquitoes happens during that 10-day period until the rabbits are dead. The less virulent strain, which still had a very high case fatality rate, 50 to 70%, but it did that death over a much longer period of time, a month or more. It transmitted to more mosquitoes through time because a longer infectious period without much reduction in the viral titers needed to infect the mosquitoes. And then later in the 50s, he isolated some what he called benign strains, and that's one of them in blue there. And it doesn't kill death, it doesn't cause much death, certainly way less than 50% of rabbits dying, but the viral titers never get high enough to infect very many mosquitoes that are feeding on them. So that was the logic that he used for explaining why the virulence had gone down, that killing the rabbits too fast truncated their transmission because of host death, and then not enough virulence meant not enough viral replication, and so the less virulent strains also did not have an advantage. And so things settled out somewhere in the middle range. And so schematically, just trying to illustrate the sorts of things he saw, this is the case fatality rate through time. And the data, when I got interested in this problem during my PhD, the data that's reported here for 1985 or so and into the 90s was based on just one or two isolates and one or two rabbits. So it really looked like what had happened was the virulence had gone down and then stabilized. And maybe there were some hints that things were changing. But when I got to Penn State, we decided that we would try to update the FENA story and see what had happened since 1980 or so. And the we here is Eddie Holmes, that many of you will know, Peter Kerr, who was a grandchild of FENA, Isabella Catadori, who is a Penn State faculty member, who's a rabbit wildlife disease biologist. And that picture at the bottom there is Peter and Isa doing work in our animal house, infecting these rabbits with the strains from the 1950s as well as the 1990s. And in the background is Dave Kennedy, who is a part of the story I'm going to tell later on. So we did that work with isolating the viruses, or using the viruses that others had isolated. And the 1990s viruses, it turned out, had changed dramatically from the viruses in the 1950s. And so on the left here is the ancestral virus, the SLS, that was released in Australia. And you can see it killing everything in about 2 weeks. On the right is one of the viruses that we were working with that was isolated in the 1990s, and it kills everything a little bit faster. So actually this particular isolate is more virulent in the laboratory rabbit than is the ancestor. So in FENA's scheme, that means we've actually still got 100% case fatality rate, but at a faster rate, so it's actually more virulent. But the amazing thing to us was that none of the original symptoms that were described are apparent in the rabbits infected with the 1990s viruses. So they don't get classic myxomatosis, which is these sort of big lesions and things. Instead, what happens is there's massive immunosuppression. So this is a classic nodule that forms up when you infect the rabbits with the original ancestor. And there's nothing at all seen with the 1990s strains. And instead, they bleed terribly. all sorts of organs in all sorts of areas. And then they have uncontrolled bacterial growth that penetrates into large numbers of vital organs. So there's rampant bacteremia without any inflammatory response to deal with them. So the rabbits have lost their ability to control their own bacteria. And there's no signs of classic myxomatosis. So we call this acute collapse syndrome, and it's very similar to a septic or toxic shock, and it seems to be associated with profound immunosuppression. And all of the 1990s viruses we looked at, all 15 of them caused this massive immunosuppression and very rapid death of the laboratory rabbit. And there's actually very little viremia involved here. It's just this overwhelming bacteremia that kills them. We also did take a look at a few European isolates from the 1990s, and they did the same thing as well. So in terms of the story, in fact, the 1990s virulence on the original rate is actually higher than the ancestor. There's a range of different death rates. The one I showed you is the fastest, but they all kill, and they're all associated. They all kill all of the laboratory rabbits, and they're all associated with this very profound immunosuppression. So the variants that's circulating in the 1990s had evolved the ability to do this massive immunosuppression. And the phylogenetic analysis says those genes arose somewhere between the end of the middle of the 1970s and the beginning of the 1980s. But there's no single gene associated with this. We sequenced all of these, and there's very different pathways to this phenotype. There are genetic differences, of course, but there's no consistent genetic variation that accounts for this immunosuppression. And there's no obvious candidate genes that are consistent across all of the isolates. There are some candidate genes that are pretty suggestive in one or two of the isolates, but those don't explain all the immunosuppression in general. So very different picture. And obviously the question is, well, why is that? Now, Fenner had also done work where he took rabbits from the field, and injected them with the standard laboratory virus. And that allowed him to track resistance in the rabbit population through time. And the general picture there is that the rabbits have become more resistant to the virus. And that's happened over the second half of the 20th century. That is actually the genetic basis of that in the rabbits is pretty well understood now, and has to do with the innate responses, particularly the innate responses early on in infection. So this is a classic arms race, if you like, where the virus is becoming more virulent. It's evolved the capacity, or the rabbits become resistant, and the virus evolves the capacity to overcome that by immunosuppression. And that looks very much like an escalating arms race. And although it hasn't been studied as well in other wildlife spillover situations, the same picture is there. If you do common garden experiments with the virus into the host, or the host standard host with viruses through time, then with those particular examples, and including that bacteria in the mycoplasma, it looks very much like all of the well-studied cases of spillover where people have been able to do these common garden cases. You get escalating virulence and escalating resistance, and it looks like an arms race that's just going upwards in all these cases. Now, we've been working a lot in the last year or three looking at updating the story into the 21st century. And these are the lineages that we've been working with. Probably too small for you to see, but some of these ones in the 1990s and some of them are after 2010. And what's most remarkable to us is that there are now 3 clear lineages that have developed. And one of the lineages is actually evolving at a faster rate than the other 2. So the rate of evolution seems to be accelerating in this lineage C. And cutting a long story short, what we see in lineage C is a further development of the clinical syndrome. So the pictures I've got there, the classic myxomatosis in panel A, that's not been seen since the 1960s. So none of the isolates that have come since the 1960s are generating classic myxomatosis in lab rabbits. The very attenuated strains is the panel D there, and none of those have been seen since the 1960s either. Everything we see is since the 1960s are of the sort between B and C. And all of the ones we've looked at have this very profound immunosuppressive phenotype, and many of the rabbits still die at great rates. But the lineages that are the isolates that we've looked at from the 21st century, some of them don't kill all of the rabbits instantly. Some of the rabbits are able to survive the immunosuppression, And those ones are associated with substantially more virus that's in the superficial tissues around the ears, the eyes, and so forth. So it's very tempting to think that what's happened is that the viruses have evolved the capacity to both immunosuppress and produce high viral titers in the peripheral tissue where mosquitoes feed and transmit the virus on. If you look at it in terms of the original FENA data, then we've got a situation where the virulence went down through the 50s, then came back up again in the 90s, and maybe more virulent than the ancestor. And now in terms of case fatality rate, it's going down, but it's associated with very, very different disease syndromes, very different clinical presentations. So just summarizing all that, since the spillover in 1950, the phenotypes, the disease phenotypes have been evolving, and there's no sign of any of that slowing down. The clinical presentation that these viruses induce is continuing to change. Very much looks like it's enhanced host resistance. So natural selection for resistance in the rabbit was met with increased virulence, and particularly the capacity to immunosuppress. In Australia today, if you look at an infected rabbit, it still looks like it's got myxomatosis, even though none of the lab rabbits do. And it seems like the combination between the increased resistance in the field and the increased virulence in the field has maintained that classic disease syndrome, but it's been caused by this balance, if you like, between the resistance and or the ongoing arms race between the resistance and the virulence. And in the laboratory, we can no longer recapture myxomatosis, the classic syndrome, except with viruses from the 1950s and 60s. So I've read a lot of the papers in the media recently when people have been talking about myxomatosis as a classic example of declining virulence. That is true, but it was a short-term effect. And obviously, if you start at 99.9% case fatality rate, there's really only one way it can go. I also think an important lesson here is that this is not about epitope evolution. We've examined all those genes and there's no change there. This is all about immunosuppression. And pox viruses are famous for their ability to immunosuppress, and this is really what's happening here. I didn't have time to show the genetic data, but there really isn't any simple genotype-phenotype map here at all. And I don't think we'd know any of this if we didn't have the experimental capacity to put viruses into a standard lab animal or lab animals to be a wild-caught host to be tested with a standard virus. Okay, now I want to turn to my second story, which involves experimental work we've been doing with a rodent malaria parasite. I've been working on this parasite for my entire career, but we've done some of that work looking at what happens when you experimentally evolve the malaria parasites through vaccinated hosts. But I just need to, before I get into that, I just need to have a comment or two on semantics here. I'm going to go forward and call this vaccine resistance. And obviously vaccine resistance has a context or a meaning in the context of human behavior. That is not what I mean. I think of this very much analogous to drug resistance, that anti-malarial, antibacterial drug resistance, that's an evolutionary acquired enhanced ability to replicate in drug-treated hosts. What I mean by vaccine resistance is an evolutionarily acquired enhanced ability to replicate in vaccinated hosts. So that's with enhanced within host fitness. Now, I recognize that some people don't feel comfortable with that definition, but I struggle a bit to find a definition which is mechanism agnostic, that doesn't imply particular types of enhanced replicative ability. So terms like vaccine escape and reductions in immunogenicity and reduced vaccine efficacy, all these things have other meanings and particular connotations and so forth. So I mean vaccine resistance in the sense of there's an evolved ability of the pathogen to replicate better in a vaccinated host. And so in SARS-CoV-2 speak, I mean vaccine resistant variants that have a replication advantage in a vaccinated host compared to their ancestors. So we might see that in reductions in certain types of vaccine efficacy, but I mean it in the sense of replication within hosts. And anybody on the call I've seen on Twitter a lot of conversation about what we call this, and I just want to say I'm happy to discuss the semantics at the end if anybody's got better terminology. Now in malaria, which of course is a great big eukaryotic parasite, evolution is actually one of the main challenges to controlling it. There's significant problems with drug-resistant evolution, significant problems with insecticide resistance in the mosquito populations that transmit it. And vaccination has been very tricky to get working in malaria, not least of which because of the antigenic variation the parasite has and the very wide range of different types of antigenically diversity in key antigens. And there's actually been vaccine trials where the vaccine has failed in real time in stage 2 trials because of changes in allele frequency. So it's been a real challenge. And although vaccines in general do not tend to suffer from evolution of vaccine resistance, nonetheless, there's a lot of diseases we failed to vaccinate or develop vaccines because of this evolutionary problem. So I got very interested in this issue with experimental system, that we could actually do experimental work looking for the evolution of vaccine resistance. Looking back on it now, I wasn't interested in the fact that the lab mouse is a novel host, but it does seem highly relevant. So the parasites come from thicket rats in Africa. They're put into lab mice, and that is a novel host. And then they get better at dealing with the lab mouse through serial passage, as I'm about to show you. So in a sense, it's an experimental spillover. And we've got vaccine candidates we can use to ask what happens to the evolution of a pathogen when it's novelly recently spilled over and vaccination is becoming a key issue. So the vaccine that we've been working with involves a recombinant protein from the AMA1 gene. And this protein is normally expressed on the end of the parasite merozoite that goes into brand new red cells. So this is the section that's binding onto the new red cells prior to invasion. And the AMA1 adjuvant vaccination with the purified recombinant antigen and a commercial adjuvant does a good job in mice of blocking that invasion. This is one of the blood stage candidates, blood stage antigens in malaria. It's been in quite a few human trials over the years. And in our experiments, the controls we use are the sham vaccination, which is just the adjuvant only. And when I say commercial adjuvant, it is a commercial adjuvant. I have no idea what it is. But the protein is purified, so we know that. And so what we're sticking into the vaccine is this purified antigen plus the adjuvant, and our control is the adjuvant only. And the series of experiments I'm going to describe here were done by Vicky Barclay, and some of these experiments were truly heroic, and I always take my hat off her taking these things on. The first experiment I want to talk about, I'm just going to talk about 2 experiments. The first one I want to talk about is one where we serially passage the parasite from a single clone, so that's one single ancestral parasite, is then passaged by syringe from mouse to mouse to mouse for 30 weeks. So every week we'd passage it on. And so it's gone through 30 mice before we've got the derived line. And then we put the ancestor and the derived lines into mice and see how they get on, and the mice are vaccinated or not. So this is the parasite dynamics through time in a series of mice. You just look at the blue lines first. So the blue line is the ancestral parasite. Here, this is the ancestral parasite in unvaccinated mice, and here in vaccinated mice. And so you can see that the vaccine is suppressing the titer. This blue line is much lower than this blue line, and it's having an impact on the initial growth rate. So this is getting up to much higher initially, and then the growth rate is sort of starved up here. So the vaccine is doing a good job of suppressing parasite replication. If we then look at the derived parasite, do the same thing, you can see that the red lines, the vaccine is reducing the initial growth rate, but not the total densities by about 10 days or 2 weeks. And so this derived parasite, the one that has got better and better at dealing with mice, so remember these mice are not vaccinated, they're just regular mice. The parasite has adapted to becoming better at mice, and that means it is better able to overcome the vaccine than its ancestor. So in my head, that is what I mean by vaccine resistance. And vaccine resistance here has evolved in the absence of vaccination. If you just look at the totals of the parasites here, that's the areas under these curves, you can see that vaccination suppresses the ancestral parasite much more than it does suppress the derived one, and the derived one has got a bit better in unvaccinated animals and quite a bit better in vaccinated ones. So the vaccine resistance is coming along as a consequence of adaptation to a new host. And in my head, that would be equivalent to the SARS-CoV-2 variants just getting better and better at humans And so we see that often as a reduction in vaccine efficacy, but it could be a simple side effect of just getting better at humans. It's harder and harder for a given antibody titer to control these parasites, the malaria parasites, given that they're better and better able to grow in a mouse. Then we did, and again, the we here is actually Vicky, what I think of as one of the craziest experiments we ever did. So this is a 400 mouse experiment, but what happened was we started again with the ancestral parasite, single ancestral parasite, and then we evolved it in lines with mice which are just adjuvant treated or vaccinated treated. And each of these is a generation of passage. We started off with 5 lines, and we kept trying to keep the lines in duplicate, but maintaining things as they went along, every week passaging the mouse on. And the red dots are where we lost parasites. So we couldn't find enough parasites to transmit on to the next week. So you can see where the mice aren't immunized, we have pretty much no problems keeping the 5 lines going. But with the vaccinated ones, because the vaccines are suppressing down the parasite densities, it's very hard to find enough parasites to get into the next week. So we have to recreate the lines and recreate the lines. And you can see that after about 10 or 12 passages, it became much easier to keep things going. So on the face of it, it looks like what's happened here is that the parasite has adapted the ability to grow in vaccinated mice. And then if you just look at the crude data through time, so this is the parasite densities at the time of passage, so when we moved them on, so it's not this is just the experimental evolution is proceeding here. The vaccine initially, that's the red dots, the parasites in vaccinated animals are lower than the parasite densities in the unvaccinated animals. But through time, those converge. And so the parasite is getting better and better able to grow in vaccinated hosts. And in fact, this difference is not statistically significant. So what's happened over time is that the anti-parasite protection, the ability of the vaccine to downregulate parasite growth, has degraded as the evolution has proceeded. So there's a degradation in the anti-parasite effect through time. The animals that were vaccinated were always better off health-wise. So there was some reduction in the animal health induced by the vaccination, but not much. And vaccination was always reducing severe disease in this case. We published this a while back now, and a journalist at the time pointed out to me, or asked me, has anybody else ever done experimental evolution in vivo of looking to see whether pathogens can evolve around vaccination? And I don't know of any other experimental evolution. I think this was the first, so I'd be very interested in hearing if anybody else knows of places where it's been done. It is amazing. There's a whole industry of people doing in vivo experimental evolution of drug resistance, but experimental evolution of vaccine resistance, I don't know of any other examples. And then if we take the derived lines and the ancestral lines, the derived lines, the ones that have gone through the sham vaccination hosts, or the derived lines that have gone through the vaccinated hosts, and then put them into vaccinated or unvaccinated animals, this is what we see. So density through time again. The top lines, you can see there's a slight difference between the ones, maybe a slight difference between the ones that have evolved in vaccinated hosts and unvaccinated hosts in their performance in unvaccinated hosts. But in the vaccinated hosts, the vaccine suppresses replication still, but it suppresses it more for the ones that have been evolving in the absence of vaccination. So the vaccinated evolved lines do better than the control lines. And if you look over here, you can see again that the vaccination is still suppressing the numbers, but it's suppressing it less so for the ones that have evolved in vaccinated hosts than in unvaccinated hosts. And really critical, we sequenced, and the details don't matter here. This is just the sequence of the target locus, the AMA1 antigen. This is the gene that produces the protein that's in the vaccine. And there was absolutely no change in this gene at all in any of the lines. And if there were changes that we couldn't detect, we think they were less than 20% frequency. So no point mutations whatsoever that we discovered in the gene that was the target of the immunity induced by the vaccination. So the vaccine is working against this protein, but this protein is not evolving back in response. Something else elsewhere in the genome evolved. And 2012, it was way too expensive to do full genomes, so we don't know where the evolution took place that allowed the vaccine resistance to emerge. So the take home from that work is that evolution in experimental evolution, at least in vaccinated and unvaccinated hosts, led to improvements in this within host replication. So my definition of vaccine resistance, that vaccine resistance occurred even when evolution was proceeding in non-vaccinated hosts. So just the business of getting better at mice generated variants that were less well controlled by vaccines. I didn't show you, but it's true that the vaccine resistant variants are more virulent because when you have more enhanced parasite replication, you get more anemia. So these derived parasites all cause more virulence, both the ones derived just by passage through naive animals and through immunized animals. The vaccine always protected against severe disease despite this evolution. So it was getting less good at controlling the parasite's replication, but it always protected against severe disease and death. And I think pretty important is that the vaccine resistance was not associated with mutations in the antigenic target. Now, reviewers are all over me about all this because we just tried one type of vaccine and we just did weekly passages. We don't know what would happen if there was every 2 weeks or something where immunity is stronger after a couple of weeks in the controls. We did transmission with a syringe, not a mosquito. And of course, we've just done, one genetic clone in one type of mouse here. So we don't know how much this generalizes. But I want to say again, I'd love to see more experimental evolution work. I think there's some really interesting potential for this in Syrian hamsters with SARS-CoV-2. And the sky's the limit. One of the things I would like to see is experiments that test this idea that an immunosuppressed host can lead to the evolution over time of variants with many, many more mutations that can cause more severe or disease or immune escape. It's not obvious to me that evolving in an immunologically deficient host is going to promote the ability to escape from immunity. That's just something that needs testing. And I think that this sort of experimental evolution is one way to do it. I'd love to do those experiments myself. Okay, my third story concerns one of the very few cases that's well recorded where vaccines have failed because of the evolution of vaccine resistance. There's been several cases of this in agriculture, but not as well studied as Marek's disease virus. This is a herpes virus. It's a big DNA virus. It's shed from the feather dander, and then it's inhaled by the birds, and that's the transmission. And at least the original viruses were oncogenic, so they cause tumors, and the birds die of, if they die of tumors. It became a pretty serious problem in the poultry industry in the 1960s, and a vaccine was introduced in 1970. It's a live attenuated vaccine, a virus vaccine. The strain itself comes from a turkey, and it produced very good protection against severe disease and death for about a decade. And then it was starting to fail in the face of what we now know to be variants that were able to replicate very successfully in chickens vaccinated with the HVT vaccine. So the second generation vaccine was brought out, which controlled the problem for another decade or so until variants were starting to break through there too. And in the 1990s, a third generation vaccine, the Rispin's vaccine, was introduced. And that vaccine seems to have held. It's still just not challenged by vaccine escape variants like the previous ones. And the Rispin's vaccine is the reason that your chicken nuggets remain cheap, because you cannot house chickens in these sorts of conditions. The high densities are brilliant situations for pathogen emergence and evolution. And the Rispin's vaccine has been a very key part of the industry's successes since then. So here's a case where, just like drug resistance, vaccines are being brought in, they're very successful initially, and then they fail in the face of virus evolution. And a replacement vaccine has to be brought in. And this evolution is played out separately in various parts of the world and is perhaps one of the best documented cases of virus evolution leading to the failure of vaccines that were initially extremely highly effective. So I've been fascinated by this ever since I first heard about it around the turn of the century. And here the we is a great group of people at the Purbrook Institute in the UK that I've been collaborating with for the best part of 20 years now on the Marek's disease virus. There's not very many facilities in the world where you can work with this virus, and Purbrook in England is one of them. what we've been doing is experimentally infecting birds initially in these isolators, and later as the EU regulations changed in these floor pens. We'd experimentally infect birds and then house them with sentinels and look at transmission. And we're also able to measure the shedding rate of the virus into the environment, because we can scrape off the dust that's coming off the birds in these ventilators and the filters, and PCR for virus. So we've been doing most of our work with what we would in COVID speak, you call variants, 5 variants in vaccinated, unvaccinated birds. And this is one of these variants, a B14, survival curve in unvaccinated poultry and vaccinated birds. So over this time, they start off as chicks and they turn into pretty big birds. But over 2 months, half the unvaccinated birds infected with B14 die, and none of the vaccinated ones die. So the vaccine is very good at protecting from death. This is a more virulent strain. Most of the birds are dead in 10 days, but there's a long tail of unvaccinated birds that survive. Here, most of them are dying in 10 days, but the tail is shorter. Here, they're all dying in 10 days, and there's some death in the vaccine group. And then this one, everything is dead in less than 10 days and breakthrough in the vaccines. So these are our 5 variants we've been working with. And these were isolated from farms in Europe and in the US over the 30 years or so in the last second half of last century. We can take those 5 variants and then look at how much virus is being shed into the environment from these birds. And so this is the copies of virus that we're picking up in these filters for these viruses. This data is, the top row is what I just showed you. This is the viruses that we're seeing in the dust. And then if you do the math, you can work out how many viruses are shed per bird through time, through the course of this time span. And so for B14, this one that kills half the birds and half the unvaccinated birds in 2 months, then the total shedding per bird is much higher in the unvaccinated birds than it is from the vaccine. So the vaccine is suppressing the viral replication in the birds, and so there's less shedding going on out into the environment. The other end of the spectrum, where these highly lethal ones are, they're killing the unvaccinated birds. And so there's almost no shedding into the environment of virus from the unvaccinated birds. The vaccines keep the birds alive, and so the shedding can go on for much longer. And as a result, you get much more shedding from vaccinated birds than unvaccinated birds. So in an unvaccinated world, B14 would take over. In a vaccinated world, 675A can take over. Just to emphasize, that's because the birds have been killed by the viruses so fast, they don't have time to shed much virus into the environment. Now, we did experimental infections of birds in these settings and then co-housed them with naive sentinels. We did a lot of these experiments, and I'm just going to show one to illustrate the issues. When we do that with the 675, the nastiest strain, all of the chicks are dead by day 10 from the virus, and none of the sentinels get infected. And that's, we know, because we can PCR the dust. There's just not enough virus coming out of these birds before they die to infect these birds. But then we keep the birds alive with the first generation vaccine. So we vaccinate all these birds and then experiment and infect them. They all survive because of the vaccine, but then the sentinels are all dead. And they start dying after about 4 weeks, and they're all dead by a bit over a month. So vaccinating these birds kept these birds safe, protected these ones, but they went on then to infect the sentinels, which then died. So vaccination enables the spread of variants that are otherwise too lethal to persist. And we did this in various different conditions. This was in the incubators, but there's also in the floor pens. Maternal vaccination is a big deal in this game, and the same thing happens if you just look at the maternal vaccination. And as I'll show later on or shortly, same thing in modern commercial broilers, which are more resistant. Now, the key issue here is that these vaccines don't prevent replication of the virus. And so this is the virus densities in the feather follicles, which is where the virus will shed into the environment for the benign variant, that B14. And you can see that the virus replication through time is reduced by vaccination by maybe 2 or more orders of magnitude. So the virus is being suppressed by vaccination, but it's still able to, even the vaccinated birds are still able to shed virus into the environment. So these are very leaky vaccines. And then the highly virulent one, it just kills everything so fast that there's not much replication possible. Whereas in the birds that are kept alive by vaccination, there's a lot of virus can be replicated in those birds and shed into the environment. So by keeping the birds alive, vaccination makes it possible for hot strains to spread to other birds. And nonetheless, the industry is doing fine. I want to say too that that sort of failure where vaccines have failed like that is rare. So Marex is an exception. That's why it's working on it. We've done a lot of work in Pennsylvania chicken farms. This is one poultry operation near State College. This is what it looks like inside, and all this dust flying around is packed with Marex disease virus. I won't go through the details, but this is the virus that we're getting per gram of dust in the farms through time, where the white parts is where the chickens are in growing, and then the grey is where they've gone off to the supermarket. And the virus, this is the virus titers through time, and you can see there's, times it was high, times it was low. The virus is there transmitting, but in the broiler operation, these are the birds we eat, the vaccines are protecting them enough to keep going on. And if there ever starts to be losses, then you use the third generation vaccine and it completely prevents things. And so they can live with this arrangement. And indeed, this is the production, or this is the disease condemnations, it's called, through time for Marex disease. And you can see that, the first generation vaccine was associated with vast reductions in losses. Second generation vaccine, some more. Third generation vaccine holds it. So the industry is doing very well because, of course, every single bird can be vaccinated. So the take home from that story, vaccine-resistant variants undermined those first two generation vaccines, took 5 to 10 years before new vaccines were needed. Those first generation and second generation vaccines suppress viral titers but don't prevent onward transmission. Some vaccine-resistant variants are so lethal in unvaccinated birds, they can only be sustained in vaccinated populations. I think it's not irrelevant that those strains that we're calling very virulent, they took 20 years to appear after vaccination started. So no one predicted them and no one saw them for quite some time. And as far as we know, the third generation vaccines have solved the problem. Now, as some of you know, that story got picked up by some experts. And this is Joe Rogan talking about our paper in August of last year. You can see this has had a rather substantial number of views. The paper got picked up as a reason not to be vaccinated, and it got pretty serious. So this is the views of our paper since it came out. This is when people suddenly realized that viruses evolved, when we started to get variants occurring. And then this is the Joe Rogan effect. And now over 500,000 people have looked at our paper, and the paper is running at the moment easily in the top 100 papers of social media all time, of, if you can believe it, the 20 million papers that Altmetric followed. And that led me, there was a lot of correspondence and a huge amount of press interest. And so in the end, I had to I did try to make the argument that really and truly, evolution can happen, but that's not a reason to stop being vaccinated. And my argument then really was then, and it still is, that it makes no sense to withhold life-saving medicines out of fear of some evolution that might or might not happen, especially if it's the sort of evolution that we could solve if it did happen, as we did in the chickens, because it was solvable with third generation vaccines. But it is a question to ask, could something like this play out in SARS-CoV-2? And just bear with me for a minute while I just go back over a little bit more data. These contemporary commercial broilers, their mothers are vaccinated with the third generation vaccines. And so they come with a lot of resistance. We did experiments with vaccinating them with the first generation vaccine, but they already come pre-baked in with the maternal protection. And so they are a lot more resistant than the ones we were working with. We did 2 experiments. The experiments stopped at different times, but these are the infectious periods of the non-vaccinated, that's the solid lines, and the vaccinated, that's the dotted lines of these birds. This is how long they were shedding virus for, either until the experiment stopped or in this case, until they died. And in this situation, this setting, where these birds are naturally acquiring the infections, the vaccination prolongs the shedding period, the infectious period, by about 2 weeks. So immunization is relaxing the selection against these variants. And variants which are capable of killing the hosts don't kill them as fast, and so there's a longer transmission period. So whether or not the virulent variants have an advantage, they've got less of a disadvantage in vaccinated hosts. Now, obviously in SARS-CoV-2, none of the death is irrelevant to the fitness of the virus. When somebody's dying of SARS-CoV-2 infection, it's long after the transmission window usually, and a fraction of it, pretty much by that point, people are either immune or they've cleared it, or they're the majority of the transmission has already happened. So death is irrelevant, at least at the moment, in the fitness of the virus. But if you just ignore for a minute the timelines here, and just imagine that each of these crosses was when a chicken felt a bit sick and stayed home from work, didn't go to restaurants, chose not to go to movie theaters or concerts, that would have the same effect as truncating the infectious period. And so I can't help but wonder if disease severity could still be an important determinant of viral fitness, playing out in terms of individuals' decisions as to whether to go into the community and infect, and whether therefore infect others. And when I look at the literature and the data, I mean, it's not very good at the moment, but it looks to me like you could make an argument that perhaps half of the transmission potential, maybe up to half the transmission potential, is if symptoms are going to appear, there's half the transmission potential could be after symptoms have appeared. So if symptoms are affecting people's decisions to go out into the world, and then we get rid of those symptoms with vaccines, then I see I've got these labeled around the wrong way. That's terrible. Sorry for that confusion. The vaccination prolongs the shedding period than the non-vaccinated. Sorry about that confusion. Nonetheless, if the individuals are decisions about getting out into the environment are being affected by symptoms, and we then reduce those symptoms by vaccination, which is the aim of the game, then we could get a situation where vaccination would allow the circulations of variants that are causing more disease, severe disease, which otherwise would not have the same transmission potential. I want to emphasize again, that's not an argument against vaccination and boosting, of course. Saving lives matters, and I still think that we shouldn't be getting hung up about evolutionary scenarios as a reason not to do something now. And it's also the case that natural immunity could do the same sort of thing. But if it happened, then obviously the unvaccinated and immunosuppressed would be at greater risk. Overall disease burdens would depend on the magnitude of the transmission advantage and disease severity immunized. As we know from Omicron, apparently less virulent variant can still cause a lot more disease if it's got a high transmission advantage. So yeah, it's very hard to know what would the effect of this would be on the overall burdens. I do think it'd be a catastrophe, at least in the US, politically, if it turned out that the vaccination was tipping the balance to more virulent variants. But in any case, I would say that this possibility is yet another very good reason to develop vaccines with substantial transmission blocking capability, which we don't have apparently at the moment. Transmission blocking rather than disease blocking. So overall, I want to just say those are the summaries of what I've just said. In terms of future variants of SARS-2, well, as we all know, its prediction is very tricky. Even extrapolation from the previous variants we've seen to try to predict what might happen is very tricky. So I don't know whether the extrapolation from other pathogens is helpful or hindrance. I do notice many people extrapolating from flu, and I just want to say I think there are other systems that we could also start to try to draw lessons from. My feeling is that we should hope for the best, but we should plan for the worst, and that planning means thinking through the very many different evolutionary trajectories that are possible, and I've mentioned some of them, but I think there are many others, some good and some bad. But I do think that we can learn, or at least get informed by the sorts of things that we've seen in other settings. And with that, I will stop.
Awesome. Thank you very much for that amazing talk. Sorry, I was trying to unmute myself. Professor Reed. Okay, so we have some time for a couple questions. We'll try our best to accommodate as many questions as we can. However, if your question does not get covered, please feel free to reach out to our speaker directly. Remember, submit any of your questions to Siang via the chat function. Okay, so I know that we have a couple of questions. Let me pull up one of them. So James was asking, in the mouse model malaria passaging experiments, you were interested in the dynamics of the parasite load given that the infection occurred. And he mentions, I think you touched on this in the limitations, but was there any evidence that ease of inoculation differed between the derived and ancestral variant? Was it easier to inoculate vaccinated mice with the derived rather than the ancestral variant?
Yeah, that's right. In both the case, whether they've evolved through vaccinated or unvaccinated hosts, it's the initial growth rate is enhanced, so they get going faster, and they in an unvaccinated mouse, they get growing faster and they go to higher levels. Yeah, so they are actually better from the get-go.
Cool. Xiang, would you like to ask your question or questions? If you want to unmute yourself. Maybe not. Okay. Well, I have a question in the meantime. So I was also interested about that malaria passage experiment because it's super cool. And we know that transmissibility plays a major factor. And so I guess I was wondering if you could maybe speculate on some of the changes you would expect had it not been, a more natural rather than inoculant sort of passage.
Yeah, obviously we skipped both the mosquito and the liver stage in those experiments. We, in other experiments, not those ones, in other experiments, we looked at the production of transmission stages, and those are the number of those. So those are the stages that infect the mosquitoes. And the number of those is directly related to the replication of the asexual stage in the host. So more asexual parasites, more gametocytes in our hands. And so that means, I think, that within host replication advantage, what I was calling the vaccine resistance, that would result in more infections in mosquitoes. Obviously, we don't know what happens to the bottleneck going from the mosquito back to the host, vertebrate host. And so it might be that there's bottlenecking there that would slow the rate of evolution down. And we do see that in other systems, that bottlenecking can slow evolution down. I think apart from the logistic complexity, that might also mean that you wouldn't see something on the short term of an experiment that would still play out in the field. So I could imagine it going either way. For me, for my money, we just need a lot more of that sort of experimental evolution work in immunized hosts.
Agreed. Super fascinating. So we've got maybe time for one or two more questions. So I'm going to ask for, I'm going to butcher this, apologies, Thai-Mui-Pham. And so they were asking, all these examples seem very specific to animal diseases and maybe perhaps exceptions rather than the rule. How likely is vaccine resistance and increased virulence due to vaccination for vaccines against respiratory tract infections? For the discussion on SARS-CoV-2, should we not extrapolate from other coronaviruses or influenza, perhaps? So, yeah.
Yeah, I do think it's a really interesting philosophical question about what's safe to extract from and what's not. And my sense is that people are, depending on your background, if you're a virologist, often the viral taxonomy is the thing you think is the critical determinant of what's going to happen. If you're an epidemiologist or an evolutionary biologist, you think about the determinants of the fitness profile of transmission as being more key. So yeah, sure, we could have ourselves a situation where this becomes flu-like, where broadly speaking, at least within a HN combination, we don't see much changes of virulence and we get antigenic drift and shift happening. Or it could be that we see more like what we've seen so far in SARS-CoV-2, that we've got these sort of what look like very much like selective sweeps of particular variants, replacing them. Not maybe with the Omicron, there's a large element of the epitope variation, but also clearly they had a lot of interesting other features to do with how they were replicating, where they were replicating, how fast and so forth. So, I think we're in new territory here about what's the key issue. Is it that you can't extrapolate across systems if it's from respiratory to non-respiratory? Is it that farms are somehow radically different from human cases? I mean, I would have said previous to SARS-2 that one of the major differences is that most of the human vaccines are extremely good at stopping transmission. That's the point of herd immunity, and that's why we got such great success with smallpox, right? If we had the levels of transmission through vaccinated people in smallpox that we do with SARS-CoV-2, would have been a completely different game. And in agriculture, of course, people is a bit more like the Wild West. And so people can really get stuck in and try all sorts of different vaccines. But, I see signs, it's not without controversy, but I see signs of evolution, vaccine-driven evolution in Bordetella, in whooping cough. There are alleles that are rising and falling in whooping cough. That's a respiratory disease. So I do the virulence genes, the toxins, as well as epitopes. So, I think it's very early days here. And I would have said if these first generation COVID vaccines were as good as the out of the gate smallpox vaccine, we wouldn't be having this conversation. But the very significant amounts of transmission that's happening through vaccinated and naturally infected people means we have now got game on for evolution. And I think the jury's out as to whether it's going to turn out to be more flu-like or, more Marek's-like or more Bordetella-like, et cetera, et cetera. So.
Gotcha. Awesome. Well, unfortunately, we are at the top of the hour. There are more questions, but on behalf of the Center of Communicable Disease Dynamics and the Harvard T.H. Chan School of Public Health, let's thank Professor Reed again for joining us today and for a really awesome talk and a fantastic discussion. Please do get in touch with any of the seminar organizers if you have any questions for our speaker, any additional questions, and also check out the CCDD website to stay up to date on the upcoming seminars and other events. Next week's seminar will be given by Professor Jessica Metcalf from Princeton University, and she'll be talking about the evolutionary ecology of coronaviruses. So thank you again for joining. Goodbye, everybody.